Okay, so welcome everyone. We're going to get started. We have a busy agenda. So my name is Montse Garcia Closas. I'm from the National Cancer Institute in the US. I'm speaking here in the morning in Washington, DC. And it's really a pleasure for me to get to welcome all of you and start the Symposium of Breast Cancer Epidemiology in Ghana. This it's a symposium that has been on the works for many years. It's one of those meetings that was supposed to be in person before the pandemic, and we've delayed as we move to this virtual world. But we hope we will still accomplish our goals of the meeting, which are to bring together all the experts and people interested in cancer epidemiology to review the current landscape and gaps in knowledge in breast cancer epidemiology in Ghana, and really build a collaborative network that we're hoping will derive into more research and initiatives to address cancer epidemiology in Ghana. And we'll do that also through exchange of knowledge and hopefully new opportunities for early career investigators that are interested in working in this important area of research. So I will now pass the baton to Janine Figueroa from the University of Edinburgh, who has been key in organizing this symposium over the last couple of years. So, Janine.
Thanks, Monsi. I just wanted to first thank the entire planning committee, both in the US, UK for me, and then of course in Ghana. This has been an extraordinary opportunity to really think about what has been going on in breast cancer research in Ghana, especially when it comes to epidemiology. So I won't say everybody's name, but this is the planning committee, and we are extremely grateful for all the input that you've made and what will, I think, be apparent to be a successful symposium today. And part of this symposium is to recognize the Ghana Breast Health Study, which was coming to fruition about 10 years ago. I was fortunate enough to be working at NCI at the time with Louise Brinton and building those collaborations with our colleagues in Ghana, especially in Komfonochi, Peace and Love Hospital, and Kolebu Hospital, as well as the University of Ghana. And so this study was a multidisciplinary population-based case control study of breast cancer to really identify risk factors, both genetic and biologic, factors, and to assess whether risk factors really differ by molecular characteristics of the tumors. And you'll hear some work today on coming from this study. And really what we're hoping is, now that it's matured, over these last few years, that this can really allow more opportunities to work together and really build some more collaborations to make sure that this has a great impact and synergizes with other efforts that are ongoing in Ghana, but also in Africa in general. So just to remind you of the setup of today's sessions, we have a welcome from Dr. Barfour first, then we'll have 5 sessions, descriptive epidemiology, molecular pathology, some selected tasks and abstracts, molecular genetic studies, and then opportunities and challenges for breast cancer research in session 5, which is going to focus really on earlier detection and treatment. And again, I just want to thank in the beginning, all the speakers and committee members and moderators for all your efforts, because I know what is going to be the next few hours, a really fantastic and interesting agenda. So with that, I would like to introduce Dr. Barfour. Hopefully I can stop sharing. Dr. Barfour, are you on the line? There you are. So I wanted to introduce Barfour. I've known him for over 10 years now. That's kind of crazy that we've known each other that long. And he's a specialist in radiation oncology. He was the head of medical director of oncology and the medical director of Komfonochi Teaching Hospital. He's currently the special advisor to the Ministry of Health in Ghana. And so I'm really excited to hear from you, and you're welcome today. Thank you so much for being here.
Yeah, thank you, Janine, and good afternoon to you all from Ghana. Distinguished invited guests, I bring you warm greetings from the Ministry of Health. I'm personally excited that after long planning meetings, this day that seemed to be so far away, some months ago, has come, and we have gathered virtually to have this symposium. I wish to welcome you all to this meeting with the theme, Breast Cancer Epidemiology in Ghana. Breast cancer incidence is increasing in sub-Saharan Africa, including Ghana, with high mortality rates. The incidence and mortality rates of breast cancer in sub-Saharan Africa are reported to be very high as compared with high-income countries like North America and Western Europe. Several factors ranging from cultural, socio-economic challenges account for this increasing incidence and mortality. There have been lots of studies in sociocultural aspects of breast cancer in Ghana, but few studies have been performed in the area of molecular epidemiology. To combat the increasing incidence and low survival of breast cancer in Ghana, multidisciplinary research studies are needed. Population-based surveys and epidemiological studies are critical to understand the factors contributing to the increasing incidence rate of breast cancer in Ghana. One such study was the Ghana Breast Health Study, which is a case control epidemiological study of breast cancer conducted from 2013 to 2015, aimed at understanding risk factors for breast cancer in Ghanaian women, as well as potential factors associated with delays in diagnosis. This study was highly successful and revealing, leading to quite a number of scientific publications. This study was conducted through partnership and collaboration between 3 Ghanaian health facilities and United colleagues from NCI, demonstrated a clear example of Sustainable Development Goal 17, that is, forming partnerships to address challenges confronting nations. This NCI-sponsored symposium had 3 aims: to bring together experts, collaborators, and advocates to review the current landscape and identify gaps of breast cancer epidemiology in Ghana, to build new collaborative networks that can facilitate improve understanding of breast cancer risk factors, to improve prevention and control of breast cancer, increase knowledge exchange and opportunity for early career researchers interested in breast cancer epidemiology and public health in Ghana and Africa as a whole. We must all at this symposium embrace the aims of this symposium and form networks that will guide us to achieve the above aims and objectives this symposium have and beyond. We understand there will be several presentations at this symposium and want to urge full participation of all participants. Ladies and gentlemen, we are having this symposium at a time that Ministry of Health Ghana has reviewed the national health policy, defined the universal health coverage that is leaving no one behind. We have developed the universal health coverage roadmap. Ghana has reviewed non-communicable disease policy, which with much emphasis on cancers and cancer research. And the government has embarked on massive infrastructure health development. So we welcome this breast cancer epidemiological research symposium, especially into areas that will change the narrative from late-stage presentation to early-stage presentation of breast cancers that will lead to more cost-effective outcomes in prevention and control of breast cancer. In our attempt to achieve the universal health coverage targets, we need all-inclusive breast cancer research, whose findings will reflect on greater segment of the country population and, for that matter, impact on greater population of Ghana. The Ministry of Health would be happy to receive report of the outcome of this symposium that will assist in the implementation of the revised non-communicable disease policy, especially in the area of cancer research. Finally, we want to thank all planning committee members and all persons whose efforts have contributed to the organization of this important symposium. Once again, I want to welcome all of you to this symposium, and I wish all of us a successful symposium. Thank you all for your attention.
Thank you very much for this introductory. We're going to be starting now. I'd like to introduce Professor Johnson. He's from the University of Ghana. He received his medical doctor degree from University of Ghana and training in public health and health policy from the London School of Hygiene and Tropical Medicine, as well as the London School of Economics. And he also did his postdoc at the University of Michigan in the US. And he's been conducting research in health systems and health services delivery from the University of Ghana. So please, Professor Johnson.
Thank you very much. Thank you very much. I would like to share my screen.
There is no blind.
So good afternoon and welcome. This is a short talk on the WHO study on aging and adult health, which is a nationwide survey in Ghana, and highlight population level issues with some risk factors germane to the discussion we are having today. The socio-demographic characteristics of Ghana shows fertility rates and mortality rates to be falling. And the implication is that there is an upward movement of the proportion of older adults and the attendant complications as people grow older, non-communicable diseases, including cancers, and that the proportion of the older adults in Ghana is expected to increase, much as is occurring in most of sub-Saharan African countries. So briefly on the WHO study on older adults, we call it the SAGE, is generally to strengthen, gather, process, and manage data on older adults so that we inform national policy and respond to the health needs of the older adults. It is a multi-country study involving 6 countries: China, Ghana, India, Mexico, Russia, and South Africa. Data is collected in waves. So for each wave is a cross-sectional study, but for all the waves, it tends to follow individuals as much as possible over a period. So each wave interviews a nationally representative sample of adults. We put the age at 50 years so they can be followed for several years, with a smaller comparison group of those 18 to 49 years in all the countries. The first wave was in 2003. The second was in 2007, which we call wave 1. Wave 2 was in 2014. Wave 3, 2018. Data not yet in public domain, but studies have concluded, and we are about to start data collection for what we call the World Health Survey Plus 2022 in the next month or so. The SAGE, as a nationwide study, involved a questionnaire-based survey that elicits information on health, disability, subjective well-being, quality of life, social cohesion, and risk factors. It also involved performance tests such as eye tests, brisk walking, grip strength, and anthropometric measurements, including body mass index, waist to hip ratios, and such measurements that are identifying risk in the individual. And we also include biomarkers, take samples for blood, saliva, and urine in some of the weights for analysis. This is just to show how the survey is conducted, pictorial. And then a picture of the map of Ghana indicating where the enumeration areas were selected from the Ghana Statistical Service. The wave 0 was based on the 2000 population census, wave 0 and wave 1. Wave 2 and wave 3 were based on the 2010 population and housing census in Ghana. And the wave 4, that is the World Health Survey Plus, which we are going to do, will be based on the 2021 population and housing census. So this is a quite representative national level data. It is mainly to identify risk, seek for intervention that will improve the health of the older adult in a supportive health and social policy environment, as Dr. Bafo a while just mentioned. So quickly, for the purposes of this presentation, I'll touch on certain risk factors: tobacco use, alcohol consumption, intake of fruits and vegetables, physical activity, and certain morbidity and chronic conditions, as well as breastfeeding, which is not directly from the SAGE, but we believe it's important risk for breast cancer. That is from the Ghana Demographic and Health Survey. Tobacco use, generally in Ghana, the prevalence of smoking is relatively low. And in the second wave, which was 2015, the prevalence was 4.8% and was more common in men than women. Men were 3 times as likely to smoke compared to the women. But as I said, smoking rates are very low in Ghana relatively. Alcohol use also showed on the population level 3%, and it increases with those who are well educated, who are in economically gainful employment, and also those who stay in the urban areas, and it declined with increasing age. Intake of fruits and vegetables, we know the role healthy diet play in most of the etiology of non-communicable diseases. And it is interesting to tell you that in all the waves, over 2/3 of older adults are said to consume insufficient amount of fruit and vegetables. And these worsened with increasing age. So the very older adults were worse off when it comes to healthy consumption of fruit and vegetables. Physical activity, which is also a protective factor for most non-communicable diseases, relatively decreased as age increased, and it is natural. The very older persons cannot exercise or do other physically taxing activities. However, at the population level, physical activity was higher among males, was higher for rural residents, and then those in the lower income group. So the risks are concentrated in urban areas, in females, and those with higher income who may be involved in sedentary type of work, living in the cities and taking car and other mobile avenues to their places. Whereas in the rural areas, typically in Ghana, people walk several distances and are involved in physically taxing activities. Obesity and overweight, another risk factor. And it is interesting to say that in wave 1, which was 2007, 1 in 5 of every older adult Ghanaian unit will be overweight per the measurement. And then 1 in 10, that's 10%, were obese. This increased quite significantly within 10 years. By 2015, 22%. So the rate of overweight had increased and that of obesity had also increased. It is more common again among urban residents, those with higher education and socio-economic status, that is those in the highest income group. So the risks are concentrated in certain specific groups of persons. This is a graph showing few of the non-communicable diseases that may be comorbid conditions in people with other cancers and other non-communicable diseases. And The most prevalent is hypertension with all its complication, and diabetes is also there. And you can see this is between wave 2 and wave 1. And some of these are becoming even more prevalent as we would like to see what the rates are in the wave 3 and wave 4. Again, to highlight the point of clustering of risk factors in certain groups, the prevalence of non-communicable disease seem to be higher relatively in the urban areas and more common among females compared to males for both of the waves that we compared here. And the ratios there show relatively higher rates in females. This is on breastfeeding. It is interesting to know that from 1993 up to 2014, the rate of initiation of breastfeeding is on the rise, and that is gratifying to know. However, one of the key important ones, which is the exclusive breastfeeding, at least breastfeeding for 6 months without adding anything to the baby's food, is seen to be decreasing from 2008 to 2014. We would like to see what happening. We would like to see what happens in the next demographic and health surveys. These are important risk factors at the population level. We put in this slide to show what we call frailty by country, and frailty is a composite index that shows the general health status from reported health condition, from measured health condition, as well as some analysis and comparing all the 6 countries. And Ghana is somewhere in the middle. The lower the line, the better, or the more healthier the older adults in those countries and all the attendant rates. Within Ghana itself, we can see that the line is lower for males, as I indicated, that the mean health score for women or female is relatively lower than that of the males, with different social, cultural, and religious factors coming into play here. Access to healthcare need, which is important. Not all the older adults needed healthcare got the care they needed, and this worsened from 2007 as we got into 2015. And when people don't get access to healthcare, then whatever risk that they have become complicated. So important for this symposium, breast cancer screening and cervical cancer screening at the population level. For this nationally representative population of older adults, in 2007, only 3% of women had had breast screening through mammography. And in 2015, that is the wave 2, it increased marginally to 4.5. That is way, way, way low if at the national level we are to combat breast cancer and other related cancers. Similarly, for the cervical cancer, the rate was also very low in 2007 compared to 2015. So access to screening services where the risk factors may be determined before it becomes complicated, it's relatively low, and that is a policy issue that needs to be looked at. In conclusion, health risks and chronic non-communicable disease are prevalent among older adult persons in Ghana. And it is also important to know that access to preventive services such as breast and cervical cancer has been poor. And therefore, as Dr. Abafo-Ewua said, there have been several national policies, but it's always beset with the implementation and ability to get results from there. And we are happy that the government is taking a lot of steps in this direction, and this symposium will add to whatever information we need to guide decision making. We believe that wave 3 and wave 4 will give a little more insight into the trends regarding the risk factors we have identified. Thank you very much. And then the acknowledgement to the SAGE team. Thank you very much for your attention.
Thank you for an excellent talk and sharing this very important data to really monitor changes in risk factors and practices that could really inform the research and the measures that need to be taken. So thank you very much for that. So we have two questions in the chat. The first one is asking whether mammography screening, the percentage that you show are for women without any symptoms, or were those for symptomatic?
Yeah, so it is at the general population level where women were asked whether you've ever had screening for breast cancer mammography. So it is not in the risk group. That is why the proportion is relatively low. It is not a hospital-based population, and it is not in a high-risk group. It is in the general population to assess access to preventive services, which is also a key indicator of how accessible preventive services are to the general population.
Thank you. We have another question. So how can researchers or people interested in accessing the data could go about that? So how could the data from the study could be accessed by others?
Yes, the study data is in the public domain, and it can be acquired from WHO upon written request. And then you have access to the data. It's being used for national decision-making, for PhD students, for master's level, and for academic publication. It's freely accessible, only you need to get access from WHO.
Great. Thank you very much. So I think we'll move on to the next talk. So we have two speakers for the next talk. So the first one is Naomi Oti for Korle-Bu Teaching Hospital in Accra. So she's a public health practitioner and a nurse specialist in radiology at Korle-Bu. And she's had extensive training and experience in nursing, oncology, and public health, and both from Ghana as well as the Alberta Cancer Institute in Canada. And she's going to be Naomi Oti for Korle-Bu Teaching Hospital in Accra. So she's a public health practitioner and a nurse specialist in radiology at Korle-Bu. And she's had extensive training and experience in nursing, oncology, and public health, and both from Ghana as well as the Alberta Cancer Institute in Canada. And she's going to be talking together with Fred Kwane Avito. He's from the Kumasi Cancer Registry. His background is in mathematics and statistics, and he's been playing a critical role in setting up the first population-based cancer registry in Ghana. He's also a consultant in multiple registries in sub-Saharan African countries, and he's worked in many committees and services together with IARC and the National Cancer Institute. So we'll get started now with the talks. Naomi, do you want to get started?
All right. Let me share. Good afternoon to all of us. All right. As already said, I'm representing the breast cancer incidence rates, both in the two cancer registries. And I'm Naomi Ohini-Oti, as said. As we all know, it has been established facts which predict exponential growth of the burden of non-communicable disease, including cancers, and in developing countries such as Ghana, which could be due to increase in life expectancy at birth, among other factors. And we all know that if we want to implement an effective cancer control program, right from prevention to palliation, we need to have a good knowledge in the incidence and mortality arising from the cancer diseases. And if we also want to have good cancer care financing, we need accurate statistics. And so for cancer incidence and mortality estimates in Ghana has been more of institutional based for some time now. And there has been a series of attempts, but God been so good. We now have 2 cancer registries in Ghana. Right. So for Accra, there has been a series of attempts, right from, if we all know about the Beckett's lymphoma series, hematomas, have a series of attempts to be able to establish cancer registry. Even way back in the 1950s, there was, someone had to look at pathology data and was published in the British Journal of Cancer way back in. As of 2002, there was an attempt made by the Ministry of Health and that of IAC, the International Agency for Research in Cancer. They tried setting it up, but that also didn't work. And then in 2012, there was also another attempt, which came out with more of the departmental registry in radiotherapy in some wards. And that one didn't have a well-defined population at risk. The hospital attendance was used as denominator, so they didn't really sit in as a population-based registry or even an institutional. And here, it was more the registrars or the abstractors were more of public health nurses who had to go take data, cancer data as part of their routine data, right? So in 2016, there was this more of collaboration, stakeholders meeting where we had the cooperation of Kolebu Teaching Hospital, other pathology services and hospitals. And then we also had a birth and death registry and a statistical services in a well-defined catchment area, and that of University of Ghana Medical and Dental School, who came in with the support of the Stanford Cancer Institute, helped drive the establishment of Accra Cancer Registry. So with that of Kumasi, it started in 2004 as an oncology department registry, which then was expanded in 2006 to include the pathology department. Then in 2008, it became a hospital-based registry with all other departments within Komfanochi Teaching Hospital are part of taking data into that registry. Then in 2012, we then transitioned into a population-based registry. So their mode of operation, data collection is done both by active and passive means, and also in terms of data collection was done on a registration form, which is unique for both registries, and is coded and entered into a registry database. And the software used by CanRec5, which is recommended by IAC, and there we use it to store data and do basic analysis. And then data is also collected from the various laboratories and oncology departments, the various departments within the hospitals, then within the hospitals. Then we also collect data from the birth and death registry. So with both registers have a well-defined administrative structure, have advisory committees made up of most of the stakeholders and who help in the taking decisions concerning the registry. And we also have a technical committee where they usually look at the day-to-day activities in connection with the director in case, in terms of data abstraction, where there are issues with staging or when there are requests coming in, the technical committees with the director and registry managers, they are in, and we also have our registries. So this is a map of Ghana for both registries. In the middle, we have Ashanti region with Kumasi there, and down south, we have Greater Accra. There's Accra in there. And these are the catchment areas for the Greater Accra district where Accra Cancer Registry, this is their defined population, which is in relation to the population and housing census 2010. The same here with Kumasi, Greater Accra. Greater Kumasi district. So I'll rest here and let Fred continue from here. Right. Fred.
Yeah, thank you very much, Naomi. And yes, if you permit me, maybe you can just be moving the slides as I speak along. And good morning, good evening, good afternoon to everyone based on where you are. So we intentionally added this particular slide just to highlight the effort that has been made in the time frame when registration has been ongoing from the perspective of the pathologists by way of helping to diagnose breast cancer. And so if you look at the graph that's been displayed, you see that both registries are recording very high percentages of histological diagnosis of breast cancer in the two registries. It's seen that in Kumasi at some point, there are some high indications for clinical diagnosis of breast cancer. So this goes to speak to the effort that has been made by way of diagnostics. We are not there yet, but we are moving in the right direction. Next slide. So if we look at the curves of the age-specific incidence rates in Accra over the age distributions, you'd see that it starts off steadily, obviously, from the young population and then peaks somewhere in early 40s to when it's really at its peak in 60s. And then there are this downward trend towards 60 plus. Yes, these are all indications for further research to ask questions as to why it seems to be going less when the ages are going up. Not that we would expect that it should be increasing anyway. And you see that in the next slide, a similar trend is seen in Kumasi, where it's practically the same profile that's seen. So this indicates that the breast cancer coverage and diagnosis have similar profiles in the two cities that we are working with. And so in the next slide, we'll talk about some successes and achievements from both registries. In Kumasi, it was the first population-based registry to be set up in Ghana. And then it gained membership into IACS AFCRN, which is the sub-Saharan hub for registries on the continent. Over some time now, I think there have been estimates from GlobalCan reporting that cervix was the leading cause or the leading cancer site in Ghana, but with the inception of the Kumasi Cancer Registry, which contributed to a review of this mathematical extrapolation. Now, breast is now captured as the leading cancer site in Ghana. And it's the data from the registry in Kumasi has been one of the sources that has contributed to scientific publications, presentations at various conferences. For instance, the American Public Health Association. Myself and some other colleagues have done some presentations there, the ASCO's, aortic, and so on. And it's also formed the basis for several collaborative research in the city. Most notably at the moment is the City Cancer Challenge, which is aimed at improving cancer care services in the city of Kumasi. And so, Naomi, I don't know if you don't mind maybe expanding a little bit more on the successes and achievements in Accra, and then I can take over and look at the rest of the slides.
Thank you. For Accra, we've also have some successes. There have been some publications also. And one of our achievements was also getting the buy-in of the hospitals. And we also had, for the first time, we had a cancer registry symposium, which was really well attended. And also the formation of the advisory board and technical committee was something that was an achievement. As one of the registries, we has a tumor registry with it. But there has been some data that have been used for some publications that are coming up, which we will see them in the near future. Over to you, Fred.
Yes, and so if you go to the next slide, I think it highlights some of the publications that the two registries have put out there. I know there are quite a number in the pipeline as well. So Kumasi seems to have 6 publications out there already, and 2 from Accra. And I understand that the teams are both working on putting out some other publications in future. And so next slide, Naomi. So operational challenges, with all the successes that have been highlighted up there, it goes without saying that there obviously are going to be some challenges that both registries face. And I'm sure it's not just pertinent to us in Ghana, but probably in my experience working across the sub-Saharan African region especially, these are largely common issues that are faced. So there really is no registry structure in our health system. And by that, we mean the registries sit individually in their cities. You'd expect that at the national level, there will be a structure that integrates these registries into the healthcare system. And it's something that we are actively working on that I'll speak to when I get to the way forward. And the challenges to record linkage due to the migration of the various hospitals that are contributing data into the registries, now using electronic medical record system, is something that is also impacting the data collection process. Also, there is no linkage as of yet to the morphology of the cancers because the hospital information management systems are now being set up to link to the pathology lab. So hopefully in the not so distant future, we'll be able to collect some information on morphologies ever since they jumped into the electronic system. Administrative challenges, obviously getting data from some facilities, some departments, and then there is no policy in Ghana which governs or guides or protects these registrars when they're going out to collect their data from the various facilities. And so the next slide looks at some of the things we can do by way of way forward. Naomi?
Yeah, I don't know.
Okay.
Oh, sorry.
Don't worry, if you block the screen, I can share mine.
Okay, right.
Okay. Yeah, hopefully everyone can see. So by way forward, we're looking at regularizing the registry staff, which as at now, at some points were on a part-time basis. I know some efforts have been made by the Ministry of Health to employ some registrars in Kumasi, but at the moment they are being deployed to other different assignments, which is taking a chunk of their time. So hopefully when we have that structure in place, all of these things will be taken away. Also, the meeting of stakeholders, both at the city levels and at the national levels, would also help improve data coverage and collection in general. And then there is this plan that has been developed to implement a national cancer registry. And I know Dr. Bafo spoke earlier on from the ministry. We've had advanced conversations into establishing a national cancer registry. And so towards the end of the year, hopefully we'll have that up and running. So this comes with an establishment of an organogram in the Ministry of Health structure so that the registry is well situated and embedded in the health system. The sensitization exercises such as symposiums of this nature would be helpful to make noise around the country and beyond on the usefulness of registry. And that is complemented with publications and research out there to the public to highlight the use of cancer registries. Thank you very much. Happy to take any questions if there are any.
Thank you very much, Naomi and Fred, for excellent presentation and really reflecting the enormous effort and value of this of obtaining this cancer registry data, both for monitoring trends, but facilitating research, informing policies. So very impressive. Given the time and that I see that there's a very active discussion on the chat next to the last talk of this session, to be sure that we have time to cover the busy agenda we have ahead. So thank you again very much for your talk. So now we'll move on to the next, the last talk. We have 2 speakers, Dr. Berna van der Puye. She's a senior consultant at the National Center for Radiology, Oncology, and Nuclear Medicine at Korle-Bu Teaching Hospital in Accra. She has been trained in Ghana as well as in the US, Howard University here in Washington, DC, as well as the University of Chicago. And she's a renowned female scientist, has been very active in research as well as training and involved in local, regional, and international cancer organizations like Aortic, ESMO, ASCO, and WHO. And the second speaker, it's Tom Heron from the National Cancer Institute. He's a cancer epidemiologist. He received a Master of Public Health and a PhD in epidemiology from Emory University. And he did his postdoctoral training at the Harvard School of Public Health. And he's now a staff scientist at NCI. And he's conducting research in breast cancer, as well as managing the breast cancer health study that Janine mentioned at the beginning, as well as a very large effort, consortial effort to conduct genome-wide association studies of breast cancer. So we can now get started. Berna, do you want to?
Bernie, I'll share. Yeah, thank you. Okay, good morning, afternoon, evening, everyone. As we introduce this topic, genetic testing of breast cancer in Ghana, I'll give the introduction because, as you know, we're just about getting into the mode of genetic testing in breast cancer. Next slide. Can I do it? Oh, okay, sorry. So what do we know about genetic susceptibility in breast cancer? There are genetic variants associated with breast cancer, and we numbered at least 180 in 2017. Some women actually were found to have a 3 times increased risk of breast cancer based on genetic susceptibility. We know that by age 40, 65% of women with BRCA1 and 45% of women with BRCA2 mutations, which only make up 1% of inherited genes. So a lot more is going on that we know that will develop breast cancer. But beyond BRCA, there's a lot more. There are also alterations in various genes, BRCA, BRAD51, that might increase the lifetime risk of women developing triple-negative breast cancer and overall breast cancer in Caucasians. A lot more has also been found that are linked to specific types of breast cancer, like hormone-positive breast cancer, and also for male breast cancer, which is something that we are all now interested in. We also know that why do we need this information? Because we know that healthy carriers of these mutations may need special care, special surveillance to make sure that they can live their full potential. The slide on my left is an introduction to a study, which I'll leave for Tom to tell us a little bit more about. Next slide. So what is the state of breast cancer genetic testing in Africa? We have 37 respondents we did a little study that was published in the Journal of Global Oncology. 37 respondents from 30 facilities in 21 sub-Saharan countries were surveyed on breast cancer management. Majority, 92%, were clinical oncologists. There was no genetic screening in any of the facilities. However, there was discussion of genetic counseling in 58% of respondents. Patients, caregivers somehow erroneously confuse genetic counseling with genetic testing. And we always think because of that, everything is so expensive and the whole subject is actually brushed aside and considered as very highly resource intensive. We also have the influence of culture, religion in understanding the concept of inherited genes as a cancer causation factor. Makes it a very difficult subject to understand, even for highly educated patients and caregivers. But as our health systems improve, caregivers and patients become more adept with new technologies and treatments. So where does that leave us? Next slide. We saw the study that 2018, many years ago, we got excited with this slide, showing the different types of genetic testing in the African populations. As you can see, not much is going on in West Africa, but it was mainly with BRCA, but in South Africa and North Africa did so much better with so many other genes. Next slide. Fast forward, we also have this study presented by Frimpong from Ghana, published in 2017, just about the same time as the other study, showing that there was a high penetrance of BRCA1 in patients with triple-negative breast cancer. So that opened up the floodgates that there's more than what we see to breast cancer in Africa, or mainly in Ghana as well. Next slide. So we need to understand the role of such interventions locally. What does accessibility mean for us in Africa, in Ghana? What are we going to do about it? Is it more about preventive or is it about management? The status quo might be different in other countries versus in Africa, or other continents versus Africa. We need data first, or else we really can't do anything, as there are ethnic variations that were observed in the previous studies done in other countries. So far, we only have private labs. We tend to export our specimens, and we all know the limitations with those. And also because of that, it makes it quite unaffordable. That a big limitation as well. We thought that we could do well with liquid biopsies. We all got excited, but the transportation of these liquid biopsies actually have problems as well. But we have a lot of local labs that are picking up technology quite fast. I guess I'm going to introduce one, the Yamachi Biotech, which is trying to localize, give local support to these testing. Now, our pathologists will tell you how much we always ask for additional genetic screening and biomarkers. So we are moving ahead as our health system. And we know our patients are poor, but there are some that actually can afford these treatments and don't need to travel outside to be able to afford these treatments. And we can do a lot more actually also with surveillance, which we're not doing very well with. But we know that next generation sequencing might actually reduce these bottlenecks that we have in genetic testing in Africa, and also to the research agenda for much cheaper options. Next slide. So with this slide, I'd like to introduce the next speaker, Tom, to talk about what they have found in West African populations. Thank you very much.
Hi, thank you very much, Verna. So yeah, good morning and good afternoon, everybody. So following up Verna's presentation there, I'm going to be presenting results from a study that we performed in the Ghana Breast Health Study that Jonine was speaking about earlier, in which we used targeted germline sequencing to evaluate the association between carrying rare pathogenic germline variants in breast cancer genes with breast cancer risks in women from Ghana. And then towards the end of this presentation, I'll also briefly be discussing breast cancer polygenic risk scores in relation to African ancestry populations. To date, the largest and the most comprehensive investigations that have looked at the association of pathogenic germline variants with the risk of breast cancer have been performed by the Bridges and the Carrier studies. The Bridges, the larger of these two efforts, included mostly women of European ancestry, but it did have a relatively smaller number of Asian ancestry women. And then the Carrier study, this study population was more multi-ancestry in its makeup. However, most of the women still were of European ancestry, but the study did include a relatively smaller number of African Americans in it. So this brings up the issue that to date, studies of rare pathogenic variants and breast cancer risk in African ancestry women are fairly limited. The largest being the before mentioned study from the Carriers group amongst African American women. We also have a relatively large study from Nigeria, and then we also have a small study that was performed in Uganda and Cameroon on just under 200 cases and controls. So to help address this gap in knowledge, in the Ghana Breast Health Study, among 871 cases and just over 1,500 controls, we used the same 34 gene targeted sequencing panel that was used by the large Bridges study to estimate breast cancer risk for carriers of pathogenic variants in known and suspected breast cancer genes. For today's presentation, since we do have limited time, I'm going to be focusing on the results that we generated on those genes that the Bridges study reported or found to be the most potentially clinically useful genes. These high-risk genes listed here and these moderate risk genes listed here. So to start off presenting some results from this analysis, I'm first going to be showing comparisons of the percentage of controls that carried a pathogenic variant in these high-risk genes, first in the Ghana Breast Health Study shown in blue, and in African-American women from the Carrier study shown in purple. And any of these comparisons that we found to be statistically significant, I've indicated here with the red asterisks. So in the control populations, we found the Ghana controls to carry a pathogenic variant in BRCA1 and BRCA2, that they were more common in the Ghana controls relative to the African American controls. If we look at the overall breast cancer cases, we found carriers were generally more common in Ghana compared to African American cases for BRCA1, BRCA2, and a strong suggestion for TP53. When we look at the cases by tumor estrogen receptor status, we found in the ER-positive tumors a greater percentage of BRCA2 carriers in Ghana compared to African Americans, and also a suggestion for this as well for PALB2. And then in the ER-negative tumors, we found higher carrier frequencies for TP53 in Ghana compared to African Americans, and a suggestion that PALB2 carriers were more common in African Americans compared to Ghanaian ER-negative cases. Here I'm now showing the moderate risk genes, but for these moderate risk genes, we found that the number of carriers or sample size amongst carriers were generally too small in both Ghana and African Americans to really make very meaningful individual comparisons. So on this slide here, we're showing comparisons of the relative risk estimates for associations between carrying pathogenic variants in BRCA1, BRCA2, PALB2, and PALB2 with risk of overall ER-positive and ER-negative disease. And we're showing these relative risks in the Ghana Breast Health Study in red, African Americans in purple. European and Asians, we're showing two relative risk estimates from the Bridges study. In blue, we're showing the relative risk estimates for these women who were found to be carrying pathogenic protein truncating variants. And in green, we're showing the relative risk estimates for women who were found to be carrying pathogenic missense variants. And then in this light blue here, we're showing the estimates for the multi-ethnic population from the carrier study. So here you can see, as I said, in red, these are the relative risk estimates from Ghana. And while these relative risk estimates, while you could see that they were more imprecise, which you could is indicated by the wider confidence intervals, notably compared to these, what we found compared to these other larger studies and these other ancestry populations were fairly similar relative risk estimates across these ancestry groups. For TP53, the other high-risk gene from within this group, unfortunately, we couldn't estimate odds ratios for this gene because we didn't find carriers amongst our control groups in Ghana, but we did identify 5 cases to be carrying pathogenic variants in TP53. Finally, from this work, what I would like to show are some estimated lifetime absolute risk that we generated for carriers of pathogenic variants in BRCA1, BRCA2, PALB2, and any of the combined moderate risk genes, so any of the 5 moderate risk genes. So if we look at this first pane here for overall breast cancer, we find that the overall absolute risk for carrying a pathogenic variant in PALB2 was just over 22% for overall breast cancer. For BRCA1, we estimated an absolute risk of just over 18%. For carriers of pathogenic variants in BRCA2, the absolute risk estimates were just about 10%. And for carriers of a pathogenic variant in any of the combined moderate risk genes was just over 3%. If we look at this middle pane here for ER-positive disease, we found the highest absolute risk estimates for carriers of pathogenic variants in PALB2 at just over 19%. And for ER-negative disease, the highest absolute risk estimates for carriers of pathogenic variants in BRCA1 at just over 17%. Again, before I move on, I just wanted to acknowledge again, there is some imprecision in these absolute risk estimates, which stem from those that imprecision in the odds ratio estimates that I showed on the previous slide. We do want to quickly point out that these absolute risk estimates that we find in this Ghanaian population are indeed much lower than what had been reported by the Bridges or the carrier study. So as an example, that in this pane here, we're showing absolute risk estimates that were reported for European ancestry women in the UK by the Bridges study. So if we just quickly look at this blue line here, these are the absolute risk estimates for European ancestry women who were carrying a pathogenic variant in BRCA1 in the UK. And the Bridges study reported an absolute risk estimate of about 55%. Again, in Ghana, we found this BRCA1 pathogenic variant carriers to have an absolute risk estimate of just over 18%. So since we found that the relative risk estimates in Ghana were very similar to these other ancestry populations, this difference can be explained by these lower baseline risk in Ghana compared to the UK. And as we heard from Dr. Yawson before, this likely can at least be partly explained by differences in breast cancer screening, which Dr. Yawson showed nicely is rather low in Ghana compared to the UK. So based on this research that I've just presented to you here, we find that the relative risk of carrying pathogenic variants in these breast cancer genes to be rather similar across these different ancestry populations, but the absolute risk for carriers in Ghana are clearly lower compared to carriers in other populations like the UK. Importantly, though, we do think that these absolute risk estimates that we're generating in the Ghana Breast Health Study can indeed be useful to help counsel Ghanaian women and other women from West African nations who are found to be carrying pathogenic variants in these risk genes. So now I'm just going to quickly just shift over from discussing rare germline variants that affect a relatively small percentage of the population to briefly discuss breast cancer polygenic risk scores. So just a really quick brief background for people who may not be too familiar. Polygenic risk scores are developed by combining the effects of common variants that are identified in genome-wide association studies that individually have relatively small effects on breast cancer risk. But by combining these effects together into a polygenic risk score, they can help us stratify women according to their risk of developing breast cancer. So to kind of present an idea of what this looks like, if we have a group of women who we think of as not having breast cancer, we can apply a PRS test to these women and assign each woman a risk score. This then allows us to go ahead and stratify these women according to their predicted risk of developing breast cancer. So we would have at one extreme women with relatively low risk scores and thus low predicted risk of developing breast cancer. And at the other extreme, we have women who would have high risk scores and thus high predicted risk for developing breast cancer. So in this slide now, what I'm showing is the performance in African ancestry women, about 10,000 cases and 10,000 controls, in which the performance of a polygenic risk score that has been developed in European ancestry women. So you can see in this table here that this European-derived PRS, while it's associated with breast cancer risk in African ancestry women, we're looking at here the odds ratio per PRS standard deviation, it clearly has a lower performance or lower association with risk than what we find in the European ancestry women. And this essentially gets at a very important underlying issue that there's been much more larger sample sizes of European ancestry women in breast cancer GWAS studies, and we need to increase or get larger African ancestry populations in breast cancer GWAS to be able to better identify more risk variants that are associated with risk in African ancestry women and ultimately allow us to build better performing polygenic risk scores. This brings me very quickly just to mention the Confluence project. This is a project that we're coordinating here from NCI. It's a large international project to study breast cancer genetic susceptibility in women and men of multiple ancestries. And I'm not going to read through the broad aims for the sake of time, but you can see them here. And in the Confluence project, we're making a large effort to try to increase representation of populations that have been traditionally underrepresented in breast cancer genetics research. So here on this histogram, you could see the number of self-reported African ancestry women that are participating in the project. In red are those women who are coming to the project with existing GWAS data. And in gray here, these are the women that Confluence will be generating new GWAS data. Importantly, a lot of this new GWAS data are coming from women in studies from continental Africa. So we're very fortunate to be having these studies participating in Confluence. Yeah, we're very excited by the Confluence project. We think this is going to help make important contributions to breast cancer genetic research for African ancestry women, as well as other women that have been left out of breast cancer genetics research in the past. So with that, show the acknowledgements. There are many people to thank for this. I won't name them all, but yeah, with that, I think we'll be happy to take your questions.
Thank you very much, Berna and Tom. It's great to start seeing more data that's relevant for the African ancestry populations, particularly for the Ghanaian population. Given that we are running over time, I think
challenges. My name is Nicholas. I've been introduced in the Department of Pathology, Kwame Nkrumah University of Science and Technology in Kumasi, Ghana. And I was twice head of department in Kofa Teaching Hospital. So I'm going to talk to you about molecular pathology. What is molecular pathology? In molecular pathology, we're looking at molecules, and we're trying to look at our molecules from tissue and body fluids. It's a bridge between anatomical and clinical pathology, and it's multidisciplinary. We look at molecular biology, biochemistry, proteomics, and genetics. We're adding molecular testing and traditional morphological diagnosis to enhance accuracy. And we're also having opportunity for precision medicine. Next slide. So common molecular methods in breast pathology include immunohistochemistry, polymerase chain reaction, in situ hybridization, DNA and RNA sequencing, and DNA microarray. Quantitative PCR and multiplex PCR are very important. And I'm very glad that Vena and Tom have taken us to deeper levels on molecular pathology. If you take a look at these slides, it's just hematocene and eosin slides of breast cancer. If you look at the one at the left, it's at lower level, where we have some proportion that is malignant, another proportion that is benign, and at the higher magnification on the right, there are some other features that we try to look at related to pleomorphism, mitosis, and also tubule formation. Generally, this method is not exactly accurate, and that is why we need more of molecular pathology. If you look at a previous study by Kwesi et al. On breast cancer in Accra, which was done in 2014, they analyzed more than 800 cancer cases. You look at the clinical demography and H&E morphology. The study was compared with similar literature from West Africa. Of course, most studies in West Africa at this particular level were doing similar things, and the study concluded that there is no changing pattern from previous studies. Patients still present late, histological type is a bad type, and the stage of presentation is a high stage. Justification for molecular pathology is that now in breast cases, we see a typical presentation. There are variable and overlapping histological patterns. There are usual biological behavior, and there is desire for the use of specific therapeutic intervention based on individual genetic factor. The summary is that the pattern recognition deployed in H&E and the clinical demography is no longer adequate for disease categorization. Worldwide, things are changing and people are beginning to look at molecular pathology related to breast cancer. And you look at Europe and you look at this particular study, which was carried across England, Northern Ireland, Scotland. They didn't get any data from Wales, and it's from 2007 and 1997. And you look at the age pattern, less than 50 years, the estrogen receptor positive was 90%, 50 to 70 years, 52%, and above 70 years, 29%. And if you look at 26% of these cases were detected through screening. That's quite different from what we have in Africa, where most of the time it is symptomatic. And if you look at what we have for women of African ancestry, they are more likely to be diagnosed with early onset of aggressive estrogen receptor negative breast cancer. The age is usually less. And this is the same thing that you see in UK and USA. They have higher mortality and they are poorly represented in literature. If you look at the immunohistochemistry studies in Ghana, and it is a gold standard for breast cancer diagnosis, I'll refer you to two papers. The first one is the one by Hoeni Yeboah and NSIJ, where they use ER, PR, and A2 for breast cancer diagnosis. And they found out that ER was just positive in 32 cases, PR positive in 9 cases, A2 in 11 cases, and they went to 330 cases. It simply shows that we have less estrogen receptor positivity in Ghana. And you look at a modified study by Titulo Ihene in 2021, where we used TMA blocks and we did the same study. We look at luminal A to be 90.8%, luminal B 9.9%, L2 positive to be 16%, and triple negative was still high, 54%. This slide shows you what immunohistochemistry just looks like. That is nuclear stain in estrogen receptor to the left, and membrane stain and cytoplasmic stain in A2 to the right. And these are very, very important. Next. So the question is, do we have opportunities for molecular pathology in Ghana? And I've seen a lot of people worried at the level that Tom was talking, whether it's a possibility in Ghana. Yes, it is a possibility. First, the availability of samples, clinical and community, we could get the samples. The human resources are available in Ghana. The availability of laboratory, both private and government laboratories are present, and there are support from universities and teaching hospitals. And with use of collaborative studies, which are multidisciplinary, we could get this opportunity for molecular pathology in Ghana done. And the availability of previous publications will be coming out, and symposium like this is to enhance our belief that molecular pathology is possible in Ghana, and grants for research, internal and external, is a possibility in Ghana. I will delve more on collaborative studies and talk about Ghana Breast Health Study. I call it GAB Breast. I'm a member of that study team. It's a multidisciplinary case control study in West Africa. And through this particular collaborative study, we gained a lot in Ghana. We have support for our infrastructure, both clinical and histological. There were provision of basic histological equipment. There's IAC support, and we're able to expand our antibodies. Human capacity building was enhanced. Local and internationally, there were training, there were workshops, and there was technology transfer. Now we could do immunohistochemistry conveniently in Ghana. There were grants to support research and postgraduate training, and publications were coming out from this particular study. Overall, there was improved patient care, because in our lesson today, we know that we are less important. The most important person are the patient. And through conferences like this and advocacy, it is a very good effort that these collaborative studies have provided. It didn't come without challenges, but we will not delve more on the challenges, and we will discuss the challenges with the solution right away. We had sampling errors, and then with imaging support, we were able to modify the sampling errors. The vegetative and specimen transportation was corrected. Standardization of tissue processing was acquired through the 3 centers. Poor documentation and record keeping, we still have some, but there has been a little bit of improvement. The social, cultural, and religious effect is still there. And some of our patients believe that mastectomy is the killer, not the breast cancer. And some patients who were managed in the hospital were lost to religious houses, and we couldn't get the follow-up. But above all, we need more coordination for histological services to enhance effective, precise, and timely reports. And we need to batch more cases together so that when we're presenting, we have enough cases to present, and it will reduce costs in molecular pathology studies. So you look at the 3 studies here, looking at the Figueroa et al., looking at impact of pathologic specimen quality on classified molecular subtype of breast cancer in 2013. Bosland talked about pathology capacity building in Ghana in 2015, and Louise talked about designing consultation for identifying breast cancer risk factors. And these were all things that we used to improve the study in Ghana breast. Next slide. In Ghana breast, we learned that we could use true cut instead of fine needle aspirate. Collection of sample prior to adjuvant therapy, which we were able to achieve. We made sure that tissues were fixed immediately in formalin cups, and we create extensive training for surgeons and pathologists, which has improved patient management outcome. Next slide. There was comprehensive histopathology review for the cases, and there were things compared between National Cancer Institute and Ghana. And Jonathan, can you enlarge that particular slide so that we can pick some salient things there? Okay, so if you look at it overall, 1,239 cases were used for this particular study. And in 99 cases, there were 10% tumor present. In like 238 cases, there was 10 to 39% tumor present. And in 515 cases, there were more than 40% tumor present. What this simply means is that there's a very good data from the Ghana breast study. Next slide. Okay, and we also had a correlative study in immunohistochemistry. And if you look at HER2, we have 79% in HER, 78% in HER2. PR is 65%. Of course, we know that nowadays PR is not usually very, very reliable. But if you look at the data we got from this Ghana breast study, the estrogen receptor was higher than many other studies. And this must be as a result of what we have been able to do in our design, where we're able to handle the fixative problem and code ischemia. There's a Spanish spectrum for breast pathology diagnosis and studying in Ghana. We're not limiting ourselves to ER, PR, and then 2 alone. Francis Okoko et al. In year 2021 look at P53 and NDM2 in cohort of breast cancer patients, and it's quite interesting. That study was done in Ghana and it was still in Ghana. Atamanu look at MOC1 and find it to be a poor prognostic marker. The limitation to these 2 studies is the number of the sample. Jonah talk about the tumor subtypes, which Mustafa is going to talk about next, and it's a very important study. And you just listen to Tom talking about the very germline pathogenic mutation, and that particular publication is under review. And Mustafa and Abubakar et al. Look at the tumor microenvironment. We look at lymphocytes, inflammatory infiltrates, we look at the stromal density, and we try to see the way it affects breast cancer risk factors. And these are very interesting studies coming from Ghana breast pathology. Next slide. So we need to bridge the gaps in Ghana. And our recommendation is that we look for more grants for research, and we continue to train personnel to make sure that we develop competent pathologists and surgeons, we need to reduce the landing cost of equipment and reagents in Ghana. We need to have effective patient follow-up system, and we advocate for total health insurance coverage for cancer cases. And we need to look at the use of procurement of reagents. And someone has talked about the policies, and we need to have a screening policies. Someone has talked about cancer registry, and the same thing is applicable to tissue repository. It should be regulated and it should be properly managed. And the university, research centers, and private organizations should join hands with government to develop good management for patients through molecular pathology. In summary, I'll say that opportunities for molecular pathology diagnosis and studies in Ghana, and the opportunities are abundant. Molecular pathology provides a venue for both clinician pathologists and other stakeholders to work better towards achieving improved patient care. Experience Ghana from Ghana Breast Health Study will be helpful in designing future studies, and this has already translated into improvement in our patient care. We sincerely rely on the continual support from our collaborators towards creating robust molecular pathology practice in Ghana. We acknowledge the Ghana Breast Health Study team, National Cancer Institute, Kolibua and Kofa Teaching Hospital are there, Peace and Love Hospital, University of Legon, and University of Science and Technology. Of course, we acknowledge the pathology team, Lawrence, Ernest, Steven, Sami, Emmanuel, Thomas, Mustafa, Junai, and Mosirat. You look at this picture, that is Mustafa, Lawrence, and I'm the one on the right.
Must be paid bien with one. I'm saying, maybe I'm
May I ask the participants to please make sure they mute their mics? Thank you.
Okay, and this is another picture to show that we enjoyed ourselves while doing this study. That is Steven, that is Jonah, and I'm there. And when you work with this team, it's always going to be a happy result. Next. And to the left is Kwame Nkrumah University of Science and Technology, and to the right is Konfa Teaching Hospital in Kumasi. That is where we are. And on the final note, I want to say thank you to everyone.
Okay, thank you very much, Joanne, and thank you, Nicholas, for setting the stage. So I'm Mustafa Abubakar from the Integrative Tumor Epidemiology branch of the Division of Cancer Epidemiology here at the NCI. And I'm really excited about the opportunity to share with you some of the work we have been doing within the Ghana Breast Health Study, investigating associations between risk factors, molecular characteristics, and breast cancer subtypes. And I'll also use this opportunity to speak about emerging opportunities investigating the associations between risk factors and tumor microenvironment phenotypes in this population. So like others before me have clearly mentioned, breast cancer is the most common malignancy among women worldwide, but the rates among women in sub-Saharan Africa have continued to rise, occurring mostly among younger women, and it has been estimated that there will be a doubling of the 2008 rates by the year 2030. The reasons for increasing trends are not fully understood, but like others before me have adduced, some considerations have been looked at, including changing prevalence of exposures, changing demographics, higher prevalence of some risk factors. So within the Ghana Breast Health Study, analysis have been conducted to investigate associations between risk factors and breast cancer. I'm not going to discuss the Ghana Breast Health Study design because that is something that Johnny has described, at the start of the symposium. But just to quickly mention that information on risk factors were obtained from detailed questionnaires, including information on demographic factors, lifestyle and environmental factors, as well as other risk factors, including use of skin lighteners and hair relaxers. And also, like Nicholas mentioned, biological specimens, have been collected for these patients for cases and controls, including blood, saliva, stool, formalin-fixed and paraffin-embedded tissue blocks, as well as pathology information and histological diagnosis, histologic grade, the tumor size, and other immunohistochemical markers, including ER, PR, and HER2. So just to give a quick summary of some of the published works that have looked at risk factors in relation to breast cancer in the Ghana breast health study, I'm just going to essentially provide a summary of the results that have been seen. So the paper by Brinton and colleagues in IGC in 2017 reported differences in risk factor prevalence between what is seen in Ghana and other populations, but the magnitude of associations between those risk factors and breast cancer risk were essentially the same as those observed elsewhere. And in a follow-up paper in carcinogenesis in 2018, Brinton and colleagues found, high prevalence of exposure, such as use of skin lighteners and hair relaxers, but in this study, use of hair relaxers but not skin lighteners was found to be statistically significantly associated with elevated breast cancer risk. And this is something that has also been reported in some other studies within the United States of America as well. And then a 2020 paper by Jonine and colleagues reported temporal trends, in certain reproductive factors, particularly parity, where it was shown that with successive birth cohorts, the number of children has continued to decline, but not so with the duration of breastfeeding, which is something also that has been, that was mentioned earlier by Professor Yeltsin in his talk. But one thing to consider is that although, like the first two papers that I showed were kind of, looking at breast cancer as a single entity, we need to recognize that breast cancer is not a single disease, but a heterogeneous group of diseases with distinct morphologic, molecular, genetic, as well as clinical profiles. And there are several subtypes, but this could essentially be broken down into two major subtypes, ER positive and ER negative, with ER positive tumors generally predominating. They tend to have a better prognosis than ER negative, but prognosis even within ER positive tumors tend to vary. ER negative tumors, like I said, are more aggressive, and women of African ancestry have been shown to be more likely to be diagnosed with early onset aggressive breast cancers, which are mostly ER negative. But again, like I said, you can have aggressive ER positive tumors, so this might be tumors that are either moderate or high grade, or harbor other molecular features that essentially make them aggressive. So in investigating associations between risk factors and breast cancer subtypes in this population. So this is a figure from the paper by Jonine and colleagues from 2020. And just to quickly summarize the result. So what was found here was that parity and breastfeeding appeared to be inversely associated with breast cancer, with risk of ER positive breast cancer, irrespective of age. But with regards to ER negative breast cancer, the risk seemed to differ with age, such that among women less than 50, the protective effect of parity and breastfeeding that you see in the other subtypes, is essentially lacking. So, but parity and breastfeeding still have other protective effect, for ER negative among women 50 years and above. In thinking about, the association between risk factors and tumor molecular subtypes, one of the things we have been kind of thinking about, and something that is really, I'm really interested in investigating, is what is the impact of the tumor microenvironment, and how does this contribute, to racial disparities in breast cancer? So trying to think about etiologic heterogeneity beyond ER status. And the question here essentially that I'm seeking to address is, do risk factors impact the tumor microenvironment biology in women from sub-Saharan Africa? So to do this, we're looking at two clinically relevant H&E-based tumor microenvironment phenotypes. And these are phenotypes that have been shown to be associated with prognosis in breast cancer, including the tumor-stroma ratio and the tumor-associated stromal cellular density. While the tumor-stroma ratio essentially is a quantitative measure of the ratio of extracellular matrix to the neoplastic parenchyma, the tumor-associated stromal cellular density is a morphologic proxy for the extent of chronic inflammation and wound healing response in the tumor. So to generate these metrics in the Ghana breast health study, we were fortunate to have two pathologists visit the NCI. So to the left is Dr. Lawrence Eduse, and then of course, Dr. Nicholas, who just spoke before me. And during their visit, we utilized computational pathology approaches. So essentially, this is using digital pathology as well as artificial intelligence, in this instance using machine learning to characterize tissue composition metrics on tumors. So during their visit, they assisted with development of SOPs for image QC, as well as annotation of viable tumor regions on slides. And then ultimately, they helped with the validation of the machine learning tissue classification and cell detection script, such that we were able to essentially generate data on tumor-stroma ratio as well as stromal cellular density. So in terms of the results, so first, when we looked at the association between clinical pathology characteristics and TME phenotypes, we observed several tumor characteristics to be associated with these TME phenotypes. In particular, tumor-stroma ratio, where we observed ER-positive tumors, as well as HER2-positive tumors and HER2-enriched breast cancer subtypes to be associated with higher levels of tumor-stroma ratio. But the strongest association was in relation to histologic grade for both tumor-stroma ratio and tumor-associated stromal cellular density. And when we looked at the association between risk factors and TME phenotypes, most of the associations we saw were in relation to tumor-associated stromal cellular density, but not tumor-stroma ratio. And in particular, we saw that having a positive family history of breast cancer was statistically significantly associated with higher tumor-associated stromal cellular density. We also observed significant trends in relation to age at menarche, and also a very strongly relationship between parity and breastfeeding with tumor-associated stromal cellular density, as well as relationship, statistically significant associations between increasing body size and tumor-associated stromal cellular density. Although the association between age and first live birth and tumor-associated stromal cellular density was not statistically significant in terms of the trend, but we found categories within age at first live birth and to be significantly associated with tumor-associated stromal cellular density. And these associations were independent of tumor characteristics, including ER grade and tumor size. And also when we did stratified analysis, looking at this by age as well as by categories of ER, we didn't find that there was effect modification by age or ER status, but in relation to histologic grade, we found this association, the association between body size and tumor-associated stromal cellular density to be modified by grade, such that among patients with low-grade disease, there was a positive association between an increase in body size and tumor-associated stromal cellular density, whereas among those with high-grade disease, this was a statistically significant inverse association. So in conclusion, this analysis found that several risk factors impacted tumor-associated stromal cellular density among Ghanaian breast cancer patients. So remember that tumor-associated stromal cellular density is a morphologic proxy for chronic inflammation and wound repair processes within the tumor microenvironment. And these associations that we saw with the different risk factors were essentially consistent with the associations of those risk factors with breast cancer risk, or their associations in relation to tumor heterogeneity. And we think that these findings, could have, implications for how we think about the relationship between risk factors and breast cancer development in this population, because, if you look at all the factors that have been associated with TS tumors, which is the stromal cellular density, it might be suggestive that the stromal pathway might be an important pathway of breast cancer development in this population. And also, this is the first study, at least that we are aware of, that has looked at this prognostically and clinically relevant tissue phenotypes on standard H&E in relation to clinical pathology characteristics. And some of the findings that we saw, particularly in relation to grade, ER, and HER2 status, might suggest that these markers might be clinically relevant, which is quite important because they're H&E based, and also because machine learning could be easily democratized and could become widely available to individuals, from around the world, including Sub-Saharan Africa. So just to acknowledge all of my collaborators in this project, many of whom are the same kind of collaborators from the NCR, University of Edinburgh, and from Ghana, Peace and Love, University of Ghana, Komfanochi, and Kolebu. And yeah, thank you.
Hi everyone, my name is Waroiro, and I'm going to be presenting my talk. The title is Do Population Structure Differences Explain the Younger Age at Breast Cancer Diagnosis in Ghana compared to the United States? So just a little bit of background, breast cancer patients in sub-Saharan African countries like Ghana tend to be younger compared to patients in North American countries like the United States. And several hypotheses have been put forward for this explanation, including biological differences, population age structure, and screening differences between these countries. So in our study, we wanted to examine the impact of population age structure on the mean age of breast cancer diagnosis in Ghana and the US. And then in a very exploratory manner, we also wanted to look at screening. We used two data sources, the Kumasi Cancer registry, which Fred and his team have very well described, and we focused on 2012 to 2016 breast cancer cases. And then we also used the US registry data and focused once again on 2012 to 2016 breast cancer cases to match the Ghana cases. But for looking at screening impact, we did use the 1975 to the 1979 breast cancer cases before screening was nationally introduced in the US. So for analysis to estimate the crude mean age, to estimate the population age effect, we compare the crude and adjusted mean age of diagnosis in Ghana and the US. And the crude mean age is the sum of age at diagnosis divided by the number of cases. And then in order to remove the population age structure effect, we standardized using the World Health Organization standard population. And in this case, the adjusted mean is the mean age that Ghana and the US would have if they had the same age population distribution. So for our results, we had the Ghana mean age, the US mean age, and then the difference between Ghana and the US. And here we are showing 2012 to 2016 years breast cancer cases for Ghana and the US. We have the crude, and then we have the adjusted. And the crude mean age for Ghana was 50.3. The adjusted mean age for the US was 61.6. And then after adjusting, the adjusted mean age in Ghana was 45.6, and in Ghana, in the US it was 55.1. So this 11.3, what it's showing us is that on average, women diagnosed with breast cancer in Ghana are on average 11.3 years younger. After adjusting, this difference dropped to 9.6, which means that 15% of the difference in mean age at diagnosis between Ghana and the United States is explained by these population age structure differences. And then, as I said, exploratory, we wanted to look at the impact of screening because, as we know, there's no national screening program in Ghana. And what we did is we compared the 2012 to 2016 Ghanaian cases with the 1975 to 1979 US cases. And after adjusting for population age structure differences, the difference between Ghana and the US dropped to 8 years, meaning that both screening and population age structure explain about 29% of the age difference between Ghana and the United States. However, after accounting for population age structure differences and very roughly for screening, there's still this 8-year difference that is remaining where Ghanaian cases are on average 8 years younger. And so in conclusion and implications, we find that as much as there's only a 15% explanation of the difference by population age structure, it's essential to account for differences in population age structure when comparing age at cancer diagnosis between two different countries. There's the question that remains of what could be explaining the remaining 8 years on average. One of hypotheses is the differences in prevalence of risk factors, specifically Ghanaian older women in Ghana have a more protective breast cancer risk profile compared to older women in the United States, leading to a decreased breast cancer incidence rate among older women in Ghana compared to the United States. There's still a couple questions that are pending here and would welcome any feedback that you have. One of them is how to accurately estimate the screening effect in the United States. This is because the US Cancer Registry does not have any information on which cases are screen detected. And then the other question is whether the average age of diagnosis and incidence rates differ by breast subtype. Unfortunately, the Kumasi Cancer Registry does not yet have information on breast subtype, but this is something that we are thinking of looking into next. We welcome any questions or feedback that you may have. Thank you.
Thank you very much. I would like to remind you that if you have any questions, just go to the chat and then put it there. I've seen some responses already coming in. So we move on to our
Can you guys see the screen?
Yes.
Okay. Hi, everyone. Thank you for having me today. Okay, so DDT is an insecticide that is known as dichloryl diphenyltrichloroethane. It's a synthetic insecticide that is used in malaria endemic countries for indoor residual spraying until other suitable chemicals become available. So it's been used for a while. It's persistent in the environment and it accumulates in fatty tissues, and it can travel at long distances and found in soils and in foods. Since malaria is endemic in Ghana and affects the lives of over like 27 million inhabitants that are susceptible to malaria infection, attempts to control malaria in Ghana began in around the 1950s. In the rollback malaria movement, DDT was granted exemption for use. However, in 2008, Ghana had banned the use of DDT, even though traces of DDT are still found in things like milk or some fruits. So currently, there are 4 recommended insecticides that are used for malaria control, and these include like indoor residual spraying or long-lasting insecticide treated nets, and they include organochlorines such as DDT, pyrethroids such as permethrin, organophosphates such as malathion, and carbamates such as bendocarb. So the health effects from the exposure to persistent pollutants such as DDT are more acute in developing countries, especially in women. So Some of the side effects have been found on various reproductive and hormonal endpoints, and they examined them in women. And although the associations haven't been found, consistently with a lot of papers, they do show that there are some causal links. So for this reason, DDT is something that is imperative that we actually examine for future research to examine the impact of DDT exposure on breast cancer. So in brief, I just kind of want to talk about how we classify DDT exposure. So we use the Ghana Breast Health Study, which is a population-based case control study to examine the potential association of malaria-related DDT exposure on long-term use of malaria control products on 3 outcome variables, which were menarche, menopause, and breast cancer. DDT exposure was classified in our analysis as ever used, so yes or no. Any of the products that are commonly used for malaria control, such as the insecticide-treated nets, the repellent room spray, the repellent skin cream, and the mosquito coil. And if they didn't use it, they were classified as no. And the study participants were females. They were between the ages of 18 to 74 years of age. And cases were the females that were pathologically diagnosed with breast cancer, and controls were females that were not diagnosed with breast cancer. So due to the environmental impact of pesticides on breast cancer and the increasing use of pesticides for malaria prevention in Ghana, this study examined the potential association between pesticide exposure linked to malaria vector control. So table 1 here shows subject characteristics of the control groups and the prevalence of malaria-related DDT exposure. So the study population included 222 females who were not exposed to malaria control products and 371 females who were exposed. The controls who were exposed to malaria DDT exposure had greater than or equal senior secondary education, which was found significant. They drank at least 1 alcohol drink a month, and that was also found significant. And they had 1 full-term pregnancy compared to women that were not exposed. And I just wanted to note that in table 2 through 4, so the next 3 slides, we're going to show the odds ratios in the 95% confidence interval. We did that to assess the association of malaria-related DDT exposure with the 3 different outcomes, menarche, menopause, and breast cancer. And we use a binary logistic regression model to analyze these and then also adjust it for potential confounders. So since studies have shown that several environmental pollutants affect reproductive factors in females, we wanted to examine the association between malaria-related DDT exposure on these reproductive risk factors, such as menarche, as you see in this table here. So in this table here, table 2 shows malaria-related DDT exposure on early menarche, which we classified as ages 10 to 12, and we were comparing them to what we classified as normal menarche from 13 to 15 years of age by the identified risk factors. The mean menarche age for individuals around the world varies. So we had to do a literature review to see what was kind of like the mean age for menarche in Ghana. And we determined that it was about 13.66 in a lot of the papers that I was reading. So we categorized menarche as early for those who were aged 10 to 12, compared them to those then who were aged 13 to 15. So when you look here at the model on the left, we adjusted here for age, but we found here that there was a 20% increase in odds of early menarche when the women were exposed to malaria-related DDT exposure compared to women that were not exposed. And when you look at the table here on the right, we adjust for age, weight, and family history of breast cancer. The findings here show that there was a 32% increase in the odds of early menarche from age 10 to 12 when the women were exposed to malaria-related DDT exposure compared to the women who were not exposed. So unfortunately, when we ran this analysis, we didn't see that there was an association with early menarche. And in table 3, we wanted to show malaria-related DDT exposure on late menopause. We classified it as 52 or older, and we were comparing it to normal menopause, which was 51 or less. So we also had to do another literature review to look at the mean menopause age for Ghanaian women. And doing a literature review, we found a couple papers that it was estimated to be around 48 years of age. So we characterized menopause as early for those that were aged, late menopause for those that were aged 52 or older. So if you see on this graph here on the left, when we adjust for age, we said the findings show that there's a 71% increase in the odds of late menopause when the women were exposed to the malaria-related DDT exposure. And then if you look on the table on the right-hand side, you see that when we adjust for these other variables, age, weight, ever drink at least one alcohol drink, and family history of breast cancer, we see that there's an 82% increase in the odds of late menopause when women were exposed to related DDT exposure compared to when women were not exposed. So a lot of this, I think, makes sense since it's shown that lifetime exposure to estrogen is a key factor in breast cancer. So exposure to endocrine disrupting chemicals, such as DDT, might be a contributing factor to the development of breast cancer among Ghanaian women, since it delays their menopause and they're exposed to estrogen for longer periods of time. So this study supports the need to explore breast cancer inducing potential of environmental pollutants such as pesticides used for malaria control. And I think this kind of goes with the literature showing that environmental pollutants do actually have estrogenic effects that do affect mammary gland development. So studying the exposure of DDT on organochlorine pesticides would be a contributing factor that could potentially be modified so we can decrease the exposure to these pesticides, especially in younger women. So in this last table, we show pesticide exposure on breast cancer. So what we wanted to show here is that if you look on the model on the left, when we adjust for age, we see that there is a 65% increase in the odds of breast cancer when the women were exposed to malaria-related DDT exposure. And when you look on the right side, when we adjust for all these other variables such as age, weight, alcohol intake, birth control pills, and family history, we see that there's a 45% increase in breast cancer when the women are exposed to malaria-related DDT exposure. So here we show that pesticide exposure does in fact seem to potentially contribute to breast cancer. So here we just wanted to show that our findings do suggest that environmental exposure to pesticides used for malaria control may have an impact on breast cancer and reproductive factors such as menopause, resulting in delayed menopause for Ghanaian women. We know that in the literature, early menarche and late menopause are known to increase women's risk of developing breast cancer. And although our findings didn't find any association with menarche when exposed to malaria-related DDT exposure, we did see that there was a delay in menopause among these women. So this study does support the need to explore breast cancer-inducing potential of these pollutants. And we would like to also note that exposure to DDT or any other of these insecticides or pesticides used commonly in Africa areas like in countries such as Ghana should be modified or reduced so we can potentially decrease the risk of breast cancer. And that's it, if you guys have any questions.
Thank you, Naomi. I think I would encourage everyone to put their questions in the chat. I'm not sure if Warilu is ready to present now.
Yes.
On to our next presenter, and that is Amber from the Ghana Breast Health Study team. And she'll be presenting on systematic review of the risk factors associations with breast cancer subtypes in women in Africa, Asian, Hispanic, and European descent. Amber.
Yes, hi. Let me, so are you seeing the slide now?
Yes.
Okay, great. Hi, everyone. I'm a postdoctoral fellow at NCI, and I am very pleased to be sharing results with you today on this project that's currently in progress. We are performing a qualitative literature review to assess evidence of heterogeneity between multiple breast cancer risk factors and ER-defined breast cancer subtypes across women of European, Asian, African, and Hispanic ancestry. For the purpose of today's symposium, I'm presenting findings that aim to answer two specific questions. First, are the effects of breast cancer risk factors in African ancestry populations similar to those in other populations? And second, is there evidence of effect modification by estrogen receptor status in African ancestry populations? We looked for case control or prospective studies published between 1990 and 2018, and we're in the process of updating the search to include publications up to 2020. We prioritized review studies, meaning meta-analyses, systematic reviews, and pooled studies, but also included individual studies that were not included as part of these reviews. The inclusion criteria included papers that reported separate estimates for one or more of these racial ethnic groups. The estimates must be looking at least one of these risk factors of interest and its association to breast cancer subtypes. The subtypes include ER positive and negative disease. If an estimate was provided for triple negative, this was preferred. Among the studies that met our inclusion criteria, the vast majority were of women of European descent. This was followed by Asian, African, and then Hispanic. As the review is still in progress, for this presentation, I'll only be discussing results for these highlighted risk factors. This table summarizes the findings by risk factor. The first question, are the risk factor effects in African populations similar to those in other populations, is represented in the first column. And the second question, is there evidence of subtype heterogeneity in African populations, is represented in the second column. A check mark means yes, an X means no, and the hatched lines indicate that there wasn't enough data on cases of African ancestry to evaluate the question. There were 3 risk factors where the answer to the first question is no, meaning that there were some differences observed in the risk factor effects in African populations compared to those in other populations. These are for number of births, age at menarche, and postmenopausal BMI. I'll give more detail on a couple of these risk factors shortly. There were a couple of risk factors where there just wasn't enough published data to assess these questions, specifically for oral contraceptive use and waist to hip ratio. Now, among risk factors where the effects in African populations were similar to those in other populations, there was evidence of subtype heterogeneity for breastfeeding duration, premenopausal BMI, and use of combined estrogen and progesterone HRT. And there was no evidence of subtype heterogeneity for age at first birth, family history of breast cancer, or use of estrogen-only HRT. Now I'll show you some data from risk factors where we saw a different pattern for cases of African ancestry, starting with number of births. To orient you to these figures, the effects for ER positive disease are on the left and ER negative disease on the right. The x-axis shows the effect estimate and 95% confidence interval. The studies are grouped on the y-axis by ancestry group. European studies in red, African in blue, Asian in green, and Hispanic in purple. We grouped estimates together that used similar exposure definitions, with the key for letters A through D shown in the footnote. Studies marked with an asterisk are the review studies. Higher parity is protective against ER positive disease, and this appears consistent across populations. There seems to be no effect of parity on risk of ER negative disease in European populations, whereas among African populations, the studies generally show a small increased risk of ER negative disease with higher parity. It's important to note that there is sometimes more heterogeneity within populations than between populations. The effects for ER negative disease among European studies is a prime example of this within group heterogeneity. Next, I'm showing the results for postmenopausal BMI. Higher BMI is associated with an increased risk of ER positive disease, and this appears consistent across populations. The effect among ER negatives is similar to that among ER positives for European and Asian populations, whereas among African populations, we have one large pooled study that shows that higher BMI is protective against ER negative disease. It's possible that the effect of postmenopausal BMI among ER negatives is different in African populations compared to other populations, and we need more studies to look at that question. To summarize, our conclusions so far are that there is often more heterogeneity within populations than between populations. Besides a few exceptions, the effects in African ancestry populations are similar to those in other populations. Having said that, more data is needed on risk factor effects by subtype in African ancestry populations. There are several limitations to this review that I want to point out. There were often differences in the exposure definitions across studies, which sometimes made cross-study comparisons difficult. There were also differences in the subtype definitions across studies. For example, some studies reported effects for ER positive disease, while others reported for hormone receptor positive disease. And lastly, the majority of studies in African ancestry populations included only African American cases. It will be very important to increase the studies of indigenous African women. And I just want to quickly acknowledge all of those that were involved in this review, and thank you for listening.
Thank you very much, Amber. We'll move on to our last presenter for this session.
Okay, sounds good. Thank you for that introduction. My name is Ebony Butler. I'm currently an assistant professor at the University of North Carolina at Chapel Hill, and was previously a postdoc at the National Cancer Institute, where I started this work and are continuing to flesh it out now. So as mentioned, I'll be speaking on reproductive risk factors in relation to breast cancer molecular subtypes in the Ghana Breast Health Study. So our goal will be to compare the exposure histories of cases and controls in GBHS to estimate the associations between 4 selected reproductive risk factors and the molecular subtypes. So as a brief background, molecular subtypes are based on RNA gene expression. The initial studies where subtypes were identified through microarray studies of thousands of genes. And what's important to note is that the genes included in these expression studies are intrinsic to breast cancer, but are differentially expressed between tumors. So for example, ESR1, we would be expected to be intrinsic to breast cancer, but may be differentially expressed lower in some tumors related to perhaps an ER negative subtype and higher in the ER positive IHC subtypes. So large-scale molecular epidemiology studies of disease heterogeneity typically rely on IHC markers. And while these markers are good proxies for us to identify molecular subtype, they lack the precision we need to identify the true intrinsic subtypes. So a brief background, in the pie chart here, I show the 3 typical IHC markers that are collected as clinical indicators for women diagnosed with breast cancer: ER, estrogen receptor, PR, progesterone receptor, and HER2, human epidermal growth factor receptor 2. So when we see expression of ER and PR, these are hormone positive tumors, and they correlate to the luminal A or luminal B intrinsic subtype. So from this green right here, you can see that we have a challenge in that ER and PR tells us something about the likelihood of the intrinsic subtype, but we're not able to distinguish between the luminal A and luminal B. The HER2-enriched tumors lack expression of hormone receptors and has HER2 amplified. And again, with the triple negative tumor that would lack expression of all 3 IHC markers, they correlate closely to the basal-like subtype or basal-like intrinsic subtype. So the method that we wanted to use for quantifying the mRNA expression needed to begin to build these subtypes in the Ghana Breast Health Study is a technology called nanostring encounter technologies. And we began with needle core biopsies of 573 invasive cases in the study. And those biopsies were processed into formalin-fixed paraffin-embedded tissue blocks. And that's important to note because oftentimes when tumors are processed in this way, sometimes you have a degradation of the nucleic acids. But what's good about nanostring and the encounter technologies is that it's optimized for fragmented mRNA found in FFPE material. Another positive is that it requires minimal input, so 50 nanograms of mRNA. And previous studies have shown that there's high correlation when you look at FFPE material matched with frozen tumor samples. So we felt very good about using this particular technology moving forward. So a brief overview of the encounter quantification system. I've included 4 steps here. And so in the dark gray circle at the bottom is our target RNAs. Again, I'll use ESR1 as an example. It's when the RNA is isolated from the tissue, it is included in a solution with a capture probe and reporter probe that are particular, or they're specifically designed to attach to this, to your target RNA. And that's shown in the cartoon to the left. The second step, the target probe complexes are purified, and then they're bound to a specialized slide or an imaging surface. And this is setting up and getting ready for the quantification step. In step 3, the slide is scanned by an automated fluorescence microscope, and the label barcodes, as you'll see here, the red, blue, green, yellow sequence. These barcodes are specific to your mRNA target. So when it's scanned by this automated fluorescence microscope, it serves as an identification for the particular target of interest. And again, just another cartoon just to emphasize the quantification here. These, in this example, we see P53, EGFR, PD1, and the color variations, again, are specialized barcodes that identify our transcript of interest. So with this particular technology and software, we are able to quantify absolute counts of mRNA transcripts that we obtain from the breast tissue. So the actual panel that we use, we quantified more than 700 individual genes on a panel called the Encounter Breast Cancer 360 panel. These genes are involved in the biology of the tumor microenvironment, and immune response. And with these 700 individual genes, you could look at a total of 48 gene signatures. In the study that I'm presenting today, we focus on a signature called PAM50, which is a 50 gene signature used to classify tumor according to molecular subtype. And so earlier I mentioned those initial microarray studies that included thousands of genes to help identify or classify breast tumors. This is a more parsimonious gene signature with only 50 that allows us to subtype tumors according to basal-like, HER2-enriched, luminal A, luminal B, and normal-like. And to the right of the screen, I included an example of an analytic data matrix. Hopefully at this stage, you're able to begin to visualize the quantification that's taking place. So on the x-axis is hypothetical patient cohort of 9 patients, and on the y-axis are genes of interest. And here with the scale here, you can see that the goal is that we want to look at the differential expression of our genes of interest across the patients included in our study.
Pick one and give me one, that's all.
So we use the, I mentioned the PAM50, the prediction analysis of microarray to classify tumors according to molecular subtype. To the right, you see the 50 genes included in the signature that are listed. And each of the 5 boxes here represents centroids that are related to the molecular subtypes. So just in short, each patient, we would look at the expression of all 50 genes. We would calculate the centroids for each of the 5, and the centroid that the patient's tumor expression patterns most closely matches is the subtype that we would assign. So as a little bit of background, I wanted to include another population-based study here in the United States to compare to the prevalence of the PAM50 molecular subtypes that we did see in the Ghana breast health study. And these data, the 2 studies were conducted around the same time, roughly the 2013 to 2015. And here we see that when looking at the normal subtype or the normal-like breast cancer, we saw similar proportions in the US and in our Ghana study. When you take a look at the luminal B subtypes, the luminal A and luminal B subtypes, I should note, are those that have are known to have a more favorable prognosis. You see higher prevalence of the luminal types in the US population, lower in the Ghanaian population. And again, that goes back to some of our discussions on screening, where screening is more likely to pick up and detect those more favorable subtypes. And so we would expect in the US that has a regular screening program to see a higher prevalence of those luminal A, luminal B subtypes. So we also wanted to see how well that PAM50 subtypes correlated with age and clinical characteristics in the Ghana breast health study. And I'll guide your attention to, the first row where we looked at the age cut point of 50. Typically in the US population, women who were under 50, we expect to see a higher prevalence of basal-like tumors. In older women, we would expect to see a higher prevalence of the luminal type. But as you can look across the different subtypes included here, we didn't see very many differences by age within subtype. We saw a slight higher proportion of luminal B tumors among younger women, but again, it roughly looks like the equivalent distribution of the subtypes by age. ER status was correlated to subtype as we might expect. For example, 80% of the basal-like tumors, basal-like cancers were ER negative, and these, the ER, PR, and HER2 status are the data obtained from clinical records. Luminal A and luminal B had higher proportions of ER positive tumors. So 82% of luminal A tumors were ER positive, 85% roughly of luminal B tumors. And again, when I'll guide your eyes to the section on grade at the last row, as would be expected, we saw that basal-like tumors tended to have the highest proportion of high-grade tumors. Luminal A, again, those better prognostic tumors had a lower proportion of the grade 3. So coming back quickly to the design of the Ghana Breast Health Study, we've discussed it in some detail, but I'll just give a few points, a few highlights that are relevant for the next few slides. So the Ghana Breast Health Study was conducted between 2013 and 2015 in 3 hospitals based in Accra and Kumasi. Eligible cases were aged 18 to 74 years, and they came into the clinic with a breast lump that was suspected as cancer. Controls were identified through census and clustered household surveys. All study participants, cases and controls were interviewed in person to collect the detailed exposure histories that I'll talk about in the next few slides. And to compare the exposure histories between cases and controls, we used polytomous logistic regression adjusted for a number of factors to control for differences between our case and control populations. So briefly looking at the table on the left, we see that the 573 cases included in this analysis tended to be slightly older. A slightly higher proportion of cases came for this nanostring analysis came from Kumasi, and similarly, similar to the higher age that we saw among cases, we saw a higher proportion of women who were postmenopausal. So this first table looks at the odds ratios and 95% confidence intervals for 4 selected reproductive risk factors in relation to breast cancer risk by molecular subtype. So the 4 risk factors of interest were age at menarche, parity, age at first birth, and median months of breastfeeding. In short, we didn't see any associations with the basal-like molecular subtype. Similarly, we didn't see associations with HER2, but when we looked at the luminal A subtype, we did see that older age at menarche was associated with a higher risk of luminal A breast cancer molecular subtype. So with age at menarche 16 or 17 and older, 85% higher and 63% higher risk. And we didn't see any associations with the luminal B subtype. And again, this is for women overall. In this table, we stratify by age. So we looked at younger women, age less than 50. No associations with the basal-like subtype, HER2 subtype, luminal A subtype. I highlight here a potential effect that we might be picking up, but it wasn't statistically significant. And so the smaller sample size here is notable, and no associations with the luminal B subtype. We saw more associations in women aged 50 and older. But again, for those hormone receptor negative subtypes, the basal-like and the HER2, we didn't see many associations, but when it came to parity, we saw a lower risk of luminal A subtype and a lower risk of the luminal B subtype with higher parity. I believe in Amber's review, she mentioned in the African populations that were included, perhaps she saw the parity was associated with lower risk of ER negative subtypes. But so here in the Ghana Breast Health Study, we see that association among luminal A and luminal B. And again, breastfeeding is an important risk factor that's been discussed throughout the day. We saw that in older women, higher levels of breastfeeding were associated with decreased risk of the luminal B breast cancer, and suggestions that they could also potentially be associated with lower risk of luminal A and HER2. But again, we're just challenged with lower sample size. So just in conclusion, the PAM50 algorithm shows strong correlation with the clinical variables in the Ghana Breast Health Study. Reproductive factors included were linked to differential risk of luminal A and luminal B type cancers, particularly among women aged 50 and older. And just one thing to note as I close, the NanoString platform allowed us to quantify more than 700 genes. Here we discuss molecular subtyping with PAM50, but it includes 48 gene signatures related to tumor genesis processes such as immune response and DNA damage. So there are many other processes that we can take a look at going forward. And thank you for your time.
Thank you.
So I think we'll now move on, and I'd like to hand over to Lily. Lily, if you are there to take the next session.
Hi, everyone. So my name is Lily Pemka from the University of Ghana. So I'm involved in breast cancer genetic studies in Ghana, but I wouldn't be talking about that today. I'm supposed to moderate the session 4, which is focusing on molecular genetic studies. Well, I've been asked to present a couple of slides on the why, put a spotlight on why we should be interested in genetic studies in Ghana and Africa in general. And yeah, I'll just have a couple of slides. Can you all see my slides? All right, so we've all got it. Yes, so we all know that breast cancer is a genetic disease, a complex genetic trait, which is caused by both genetics and then environmental factors, also known as a multifactorial disease. And with genetic diseases, it usually presents with ethnic and racial variations. And based on the previous studies, so the previous participants who have spoken today, you can tell they've highlighted certain disparities that's kind of separates people of African descent from other populations in terms of maybe the subtypes. The subtypes that are prevalent in our population, the aggressiveness and all that with breast cancer. So also there's this vast disparity when it comes to research between low and high income countries. So here I'm going to highlight a few of the gaps that are glaring when it comes to breast cancer genetic studies in our parts of the world. So not just in Ghana, but in West Africa in general. My slides. Okay, so first of all, data on breast cancer research is relatively scanty, and epidemiology is not clear. But thankfully, I'm happy to say that from following this symposium, it will be less unclear. And if you look at the genetic studies that have been conducted, both genetic and genomic studies in general, Africa is profoundly underrepresented. If you look at this image on the side, it's a study that was published on general genome-wide association studies around the world. And if you look up here, Europeans are overwhelmingly, represented at around 78%. Now, if you look at people of African descent, this is just a paltry 2% of our population represented right here. Now, because of that, because of the dearth of genetic studies, the genetic risk factor associated with breast cancer in Ghana and Africa in general, it's unknown. It has barely been scratched. And like I already mentioned, most of the studies are overwhelmingly European. And studies from those populations are hardly representative of the extant, African population. And even if you look at African Americans, and I know one of the speakers mentioned that studies from African Americans, data from African American studies are not fully representative of the extant and African population, owing to admixture and other factors. Now, because of these genetic differences, there's a need for population-based studies. Now, if you look at human migrations, modern humans evolved in Africa, and then around 100,000 years ago, they started migrating out of Africa in waves and then populated the rest of the world. And because most of the Africans remained on continental Africa, the majority of the genetic diversity is found in African populations. There's an image here which showing the haplotype heterozygosity of human populations as a function of distance from Addis Ababa. Now, if you move from Addis Ababa in Africa and you move outwards all the way to America, you can tell that the genetic diversity significantly drops. So it doesn't make sense to copy and paste, genetic or genomic data from, European populations. It really doesn't work for the continental African populations. So now, based on reviews that have been conducted, it's been shown that Africa is the most diverse, has the most diverse population genetically, and it's usually the most informative way to identify genetic risk basis of complex traits like breast cancer. So because of this high diversity, there's a unique contribution to discover variants that predispose us to certain complex genetic diseases. And there's a huge opportunity to identify novel loci. For example, Sherman and Co. in 2018 published this paper, which was really illuminating, really eye-opening, where when they looked at the sequence 910 people of African descent, so you had continental Africans who from the Caribbean and whatnot, and they were able to show that there were 10% extra bases of nucleotide bases that had never been seen and which were missing from the reference genome that is typically used for all genomic studies. So that tells you there are a lot of variants that have never been identified. So for studies of this nature, there's an overall hypothesis that shows that states that people of African descent, apart from the known variants that have been seen in African population, other populations, would have unique susceptibility loci, which would predispose them to breast cancer. And the overall goal is to identify, or present a more comprehensive genetic landscape for breast cancer in people of African descent. Hopefully that will be used to develop novel molecular detection techniques and also for therapy as well. I was happy to see that someone mentioned genetic counseling in the chat. I think is Dr. Vanderpoel, I'm not really sure, but it's a good point to raise. The good thing is the WAGMIC Center in Ghana has started the first genetic counseling program probably in West Africa. And hopefully once we're able to generate all this genetic data, it will serve as a resource for genetic counseling as well. Thank you. All right, now I'm gonna introduce the three speakers for this session. Here we have both Dr. Lisa Newman and then Dr. Melissa Davis. And they are both doctors at the Weill Cornell Hospital involved in breast cancer study that focuses on racial disparities, disparities around the world. So Dr. Lisa Newman will be talking about lessons learned from the International Breast Cancer Study, including work in Ghana. And then afterwards, we have Samuel Huno, who is
researchers on the continent of Africa to evaluate the genetics and the biology of breast cancer among women with African descent. My journey with this international work began in approximately 2004 when I was working at the University of Michigan. And because we were so interested in understanding why African American women have higher rates of triple negative breast cancer, we wanted to perform genetic work looking at women who have shared ancestry with African American women. And that led us to a very exciting partnership with our collaborators at the Comfort-Nochi Teaching Hospital in Kumasi, Ghana. We started our collaborations again back in 2004, and subsequent to that, I had the pleasure of meeting Janine because of her interest in studying breast cancer among Ghanaian women. And Janine, I thank you so very much for allowing me to work with your team in subsequent years and for inviting me to be a part of this symposium. Initially, in 2004, I met Dr. Bafor Awa, who is on this symposium. Bafor is a radiation oncologist, and we became very good friends almost immediately and established our initial partnership between the University of Michigan and Comfort-Nochi. Subsequently, we have had the pleasure of working with an incredible multidisciplinary team at Comfort-Nochi, including Dr. Ernest Ajay from Pathology, from Surgery, Dr. Francis Apilla, Dr. Ishmael Chi, Dr. Joseph Apong, for Medical Oncology, Dr. Ernesto C. Bonsu. And over those years, we've amassed a very large biorepository of breast tumor tissue and saliva and blood specimens suitable for the genetic work that we were interested in conducting. We also had opportunities to invest in the pathology services at Kumasi with establishment of immunohistochemistry programs, and to support our surgical colleagues with establishment of core needle biopsy programs so that the cancers could be diagnosed more efficiently and more rapidly. As we expanded our international work, we established partnerships in Addis Ababa, Ethiopia, based upon the St. Paul's Millennium Medical College Hospital, working in partnership with Drs. Bekele, Dr. Abedi, and our pathology colleague, Dr. Aisha Jibril. And it was through these partnerships across the different regions of Africa, comparing Western and Eastern Africans, that we started to see some very intriguing differences. And we have called these explanations for these differences a field of research that we've termed oncologic anthropology. And in particular, we've seen many similarities between the breast tumors of African American women and Western Sub-Saharan African women, which we believe is related to the shared ancestry here because of the transatlantic slave trade. And we've continued to expand, now having partnerships in Uganda, Nigeria, in the Caribbean, on the island of Antigua. And it's just been so exciting to see this work grow and to see the many friendships that we've established continue to grow in depth and in robustness. So at this point, I am really happy to turn the podium over to my research colleague and the scientific director for our team, which is now called the International Center for the Study of Breast Cancer Subtypes. And Dr. Melissa Davis, my research partner and scientific director for the ICSCCS, is going to talk to you about her fascinating basic science and genetic research based upon the biorepository that we've established since 2004. So again, I thank you for allowing me to participate.
Great, thank you, Lisa. Actually, you probably gave the first half of my talk. I was trying to follow along with you and present some of the slides. Unfortunately, I don't have a lot of time to go through all the details, but kind of falling in line with what Dr. Newman has talked to you about so far is just basically going to be a tour of all of the different projects that we're able to conduct and some of our high-level findings because of our partnerships there on the continent of Africa, particularly in Ghana. Right now, I'll give you a highlight from our annual meeting. We had our 5th annual meeting last year, which was sponsored in part by an R13 from the NCI. We have additional partnerships with other institutes and even a biopharma grant to look at specific biomarkers, particularly in our African cohort. And this is just a snapshot currently of our prospective recruitment populations. The 3 bubbles across the boroughs of Manhattan represent the New York Presbyterian hospitals. And then of course we have several sites across Africa at various stages of recruitment. One of the things I like to brag about is in our mission of capacity building, we also make sure that our collaborators benefit from the scholarship of our various publications. The ones that I'm highlighting were, again, from the update of our annual meeting and represent the publications that were published between our 2020 and 2021 meetings. So as you can imagine, trying to have an international effort really requires teamwork, seamless, whenever possible, seamless collaboration and integration of any number of different aspects to think about. This is just a snapshot. I don't have time to go through each of them, but they all cost money. So we have to be very creative as well about where we get our funding. This is sort of a workflow of the samples that we collect. As Dr. Newman mentioned, we collect germline, saliva, blood, and as well as FFPE tumor samples at all of our sites. However, we also are starting to collect fresh tissue from our Ghanaian site to generate some ex vivo organoid models. As Dr. Newman mentioned, she's very active in terms of clinical enrichment and training, providing clinical services. This is a snapshot from a recent publication where because of different pathology workshops and fellowships that Dr. Newman sponsored for the Department of Pathology there at CATH. We saw sort of an improvement over time of sort of the rater agreement, if you will, of the estrogen receptor status. And so this is actually over time, we see this progression of improvement because we're not only providing reagents, but also training and consultation for instance, diagnosing hormone receptor subtypes. And as one of the presenters mentioned earlier, having this type of information is actually kind of rare on the continent. And so we hope that, you know, our efforts can transcend just beyond our hospital site. This is a snapshot of the enrollment efforts at Confanochi. This was our year 2020. So the numbers are actually 2021. The numbers actually had a sharp decline at various points, sort of correlated with the COVID-19 spike. We also capture ethnicity background. By far, the majority of our enrollment represents the Ashanti tribe because the Confanochi Hospital is actually central to that region. And then because we share resources, so I train laboratory techs, we've even purchased laboratory equipment, and we bring laboratory equipment to the site to do blood processing. And so we incorporate, for instance, screening of infectious diseases, and we also have trained them to do biospecimen processing, including both the buffy coat and plasma cryopreservation. And then lastly, from an overview standpoint, we collect fresh tissue, but we've also trained them to be able to do this in our absence to cryopreserve viable cells, which then become part of our organoid ex vivo model system in partnership with the Englander Institute for Precision Medicine. And our lines from Ghana actually some of the best growing. I'm going to show you a bit of data here in a second. As all of you know, basically our focus is on breast cancer disparities. I don't have to spend a lot of time talking about it, but you know, the clues that sort of pointed us to the direction of biological determinants had a lot to do with the fact that white women had higher incidence, while Black women had a higher mortality or lower survival, if you will, because of the implementation of hormone-targeted therapies, which sort of unsurfaced the fact that there are different biological types of breast cancer that are more prevalent in African-American populations. This, on top of course, affluence, can certainly impact survival. But even when we look at, for instance, the breakdown of affluent versus poor counties, African-American or Black patients with prostate and breast cancer still suffer the highest mortality rates, which then again points us to some biology, which is this triple negative breast cancer. Even within triple negative breast cancer, very quickly across the hospital systems that we've been engaged in, which include Henry Ford Health Systems in Detroit, as well as New York Presbyterian Hospitals in New York City, we actually see between screen detected and non-screen detected, African Americans have a wider gap of disparity. But even looking at, for instance, the 5-year survival of the screen detected, which should be an earlier onset, more curable form of triple negative breast cancer, we still see sort of a 10% disparity in survival. And some of this could be potentially due to compatibility, if you will, of the chemotherapies that are currently first-line treatment. One of the studies that we're conducting right now is looking at the frequency of alleles that are known to have cardiotoxicity. We know, for instance, looking at the global distribution of these known alleles from pharmacogenomics, that populations of African descent actually have the highest frequency of these alleles. Almost a third, if you will, of the African diaspora have predisposition to cardiotoxicity. And so we have, for instance, as Dr. Newman mentioned, I'm only showing the bit of the cohort that we have chemo data for. And we're certainly enriched with other populations, including, for instance, people of African descent, including our Ethiopian and African patients. And we're also eager to look at our Hispanic population, because we know that certain Hispanic populations also have a large amount, if you will, of admixture from Africa. I won't spend a lot of time on this because one of the previous speakers already pointed out the dearth of genomic data, where the majority of our public understanding of genetics in general is based upon European populations, despite that portion not being the major global population. This translates into clinic with variants of unknown significance on known cancer genes. As was also mentioned, there's areas across the genome that have not even been interrogated. And so we need to improve these tools by improving the data. Dr. Newman already gave you some insight on the oncologic anthropology aspect. I'll just add that in every country in our systematic review, we found that when we could separate out the portion of the population of African descent, it was that component of the population that has the highest rate of triple negative breast cancer. And so all of this really points to a potential link related to genetic ancestry. So after joining ICS-BCS, one of the first things we did was measure genetic ancestry across the multi-ethnic cohort. And we found that West African ancestry was associated with triple negative specific breast cancer. We looked more specifically within just African Americans who have an admixture of European and African ancestry, and we still see retained that the percentage of African Americans that have the highest amount of West African ancestry also have a higher risk of triple negative breast cancer. So all of this is really pointing towards the fact that there's a shared genetic background. We know that the current or contemporary African diaspora, which is due to the transatlantic slave trade, is very unique in terms of migratory patterns. For instance, founder populations are typically hybrid populations, whereas the African diaspora actually suffered from continuous gene flow across centuries. Millions of Africans were displaced over this time. And so we are still able to see more appropriately termed comorbidity associated genes. And so African Americans, for instance, suffer from a very unique environment, and for instance, diabetes is more prevalent in this population. Sorry, I can't just skip the slide, but one of the things we identified was an immune signature, which was already talked about in another talk, which is based on what we believe to be the dichotomous function of a gene called DARK. There's a mutation in the gene called DEPI-null that regulates systemic inflammation, but also tumor microenvironment. And so we're currently funded through a couple of mechanisms to look at this African-specific DEPI-null blood group and how this, in the context of breast cancer, can alter the tumor microenvironment. And we're using a couple of platforms, including imaging mass cytometry, to do single-cell analysis as well. So again, none of this would have been possible had we not been able to partner with our African collaborators. And we are certainly looking forward to sharing the outcome of our work. Let's see. I think I can just stop here and leave room for other questions from the group. I wanted to show my acknowledgement slide. Here we go, because I do want to thank everybody who's a part of the study and our funders. So thank you for your time.
Hello, my name is Samuel Ahuno. Today I'm presenting on studying cancer genomes in Ghana using liquid biopsies. So African women have one of the highest breast cancer mortality rates in the world, largely due to the issue of late presentation. So at the time of diagnosis, a large number of patients have very big tumors, and it becomes very difficult to do the necessary intervention in time in order to save their lives. So current diagnosis relies on the doctor examining the breasts and referring these patients to one of the few tertiary institutions in the country. This is Dr. Wiafi, one of our collaborators, doing an exam on the patients at a facility. And it's also very important to note that, there are limited facilities, and sometimes it's very difficult, for women to seek help, from the limited facilities. They would have to travel from different parts, of the country, to be able to seek the necessary medical help. So we also hypothesized that differences in cancer biology might also contribute to the higher mortality in Ghana. This is also due to the fact that there is limited there is also limited data on the comprehensive mutation landscape of breast cancer patients in Ghana, and also we also didn't know much about the differences between Ghanaian breast cancer women, compared to maybe the whites and in the US, and also African Americans in the US, in terms of the cancer drivers and also other fields of the genomics. So this is a study done by Dr. Janine. So a lot of the tumors in Ghana are estrogen negative cases. A lot of them too are triple negative breast cancer cases, which is also one of the aggressive subtypes we have. So we actually need solutions that would work for all cancers, not just for breast cancer. Why? Because it is estimated that the numbers in terms of incidence and mortality would probably double in the next 20 years, and global oncology efforts are looking at ways to do early detection and also molecular epidemiology to find differences, and also build capacity of people in the SAP to be able to carry out some of the operational research and molecular research as well. So we also hypothesized that liquid biopsy offers a window of opportunity for diagnosis and also for treatment, monitoring. Now, liquid biopsy, it's basically using biologically available fluids, which could be, in this case, blood, or maybe the urine, or maybe saliva, to be able to do, or to be able to retrieve diagnostic information. So in the case of breast cancer, you could take like a small amount of blood from the patients, rather than taking a tissue biopsy from the breast, which is very invasive, very painful, and a lot of women are afraid of or scared of. You could take just a simple blood draw and be able to quantify the DNA in the blood, and be able to also quantify the DNA that could be associated to cancer, and be able to extrapolate or get your diagnostics information with regards to breast cancer, and also other types of cancers as well. So we developed this study, which is under the a bigger study initiated by the NCI, particularly looking at circulating tumor DNA for breast cancer detection. And these are really wonderful people I got a chance to work with recently. So, as I mentioned, the Ghana Breast Health Study, it's a big study initiated by the NCI in collaboration with Ghanaian institutions. It's a population-based case control study with over 2,000 cases. It was really done well with socio-demographic information collected and so many information collected to be able to really study breast cancer in detail in the country. So for this part of the talk, we did a cell-free DNA study. So this is basically an overview of the experiments and design. So from the Ghana breast health study, we got access to 15 patients with, confirmed, cases of breast cancer. And then, we had access to the blood samples. So we were able to extract cell-free DNA, and then which was, sent to the Broad Institute, for sequencing. We, did two things. We, sequenced using a relatively, cheaper, relatively inexpensive approach, which is called the 0.1 ultra low pass, sequencing, and a more standard approach, which is the 30x whole genome sequencing approach. Ideally, we'd want to use the cheaper version in Ghana, but we also want to see how best that also performs compared to the standard as well. We did some of the bioinformatics in Mount Sinai, and we also worked on the cloud. It was a really wonderful experience. Yeah. So as I was saying, we extracted, cell-free DNA from, the patients, and then we, sequenced. The idea is that, we should be able to apply to a larger cohort because it is very, cost effective. And it's also very, scalable. The methods were developed, at the Broad Institute by one of our collaborators, Dr. Gavin Howe, who is currently at the Fred Hatch Cancer Institute. We used his tool called ICOCNA to be able to estimate, tumor fraction, which is the amount of DNA which we could associate to tumor in the blood. And then we also did, copy number alterations, which is looking at, large scale genomic, aberrations or damages across the genome. So with the tool, at the initial phases, we get something like this. So this is basically plotting the read count across the genome. This is chromosome 1, 2, 3, 4, 5, up to chromosome X. You could see it's very noisy. You can't really see like a distinct pattern. So this is what we get initially. But then we, take it through several processes, of really cleaning and normalizing and correcting the data, and we get, something, beautiful like this. So this is the GC correction, phase, and then this also the mappability correction phase. So you see, as we do the correction, we are able to get, good signals to be able to, increase our confidence in saying, these regions of the DNA maybe are amplified, or maybe, they are stable, or maybe they are, deleted. Yeah. So, at the end, of everything, you get, something like this. So this is a genome-wide, plot. So the red areas is showing amplification, and the green ones are showing deletions. And this is what I did. I kind of like mapped specific regions that contains specific cancer genes, especially drivers like the MYC and the TET. And we could see, some of them are amplified. Yeah. So the key result, is that we could detect cell-free DNA using both, approaches, as I mentioned, 30x, kind of a good standard. We were able to detect, our patients. We were able to detect, cell-free DNA in all the patients using the 30x. You could see we missed, a few when we used the 0.1s, which is what we want to use, ideally, in the real world. But I don't think this is very bad. I think this is very, promising. Yeah, more than 70%. I think this is very promising. So we also compared, the tumor fraction. in our cohort to a study which was done also in the US. And so what we found was that even at the time of diagnosis, Ghanaian women have high amounts of cell-free DNA compared to US patients at the time of diagnosis. So this suggests that we have a unique situation where we could use this tool to be able to do detection in our setting. This is still a recap of the copy number plots, basically looking at the genome-wide plots, and then you could see amplifications and deletions. So these are patients with very high ctDNA fraction, about 24%, and you could see patients with very high tumor fractions. They usually have lots of chromosomal aberrations. There are a lot of amplifications, and lots of deletions as well. So patients with very low tumor fraction, we could see that the genome is fairly stable. So we did we took some information from here, then we plot specific genes, looking at their copies. So the top diagonal is for the 30X experiment, and then the bottom diagonal is the 0.1X experiment. So basically, we want to see like a high concordance between the top and the down, the 30X and the 0.1X. A lot of them, there is a high concordance in majority of them, except some few cases. We also compared ctDNA across subtypes, TNBC and non-TNBC. TNBC, triple negative breast cancers tend to have higher ctDNA, which could also be explained by the aggressiveness of the disease, but this wasn't statistically significant. Also, we also compared ctDNA across grade and also tumor size. Also, this is also not statistically significant, but then you could see like a trend where large tumors basically have very high tumor fractions. This is quite unexpected because this could also be explained, that we have very few patients with grade 2, and very and a lot of people with grade 3. So probably skews the data a little bit, but they weren't statistically significant then. But then we could see the trend. So future directions is to be able to confirm the clinical utility of liquid biopsy for diagnosis and disease management in Ghana, and to also tailor the assays for the Ghanaian population and health system, and to be able to use the cell-free DNA to predict tumor types and also survival. I would like to thank all my collaborators, especially the patients that gave us the samples, my supervisors, Dr. Polak, Dr. Kwatin, and our collaborators in Ghana as well. They did an incredible job facilitating this study. Dr. Johnny Monte, Dr. Monte as well. They've been very incredible working with, and thanks.